Now I am very, very delighted to introduce our fantastic speaker, Dr. Dil Thavarajah, who is an associate professor in the Department of Plant and Environmental Sciences at Clemson University. And also, we're very lucky and proud to have her as a GFI grantee. Dil is the head of Clemson's Pulse Biofortification and Organic Nutritional Breeding Program, co-leads the phenomics component of the Feed the Future Innovation Lab for crop improvement at Cornell, and her research primarily focuses on the genetic biofortification of lentil, field pea, and chickpea to improve human nutrition. We're so excited to have her here today to discuss how plant breeding can optimize the protein quality and bioavailability of pulses, specifically for plant-based meat applications. So a very underexplored research area, and I can't wait to learn from you today. So Dil, whenever you're ready, you can go ahead and share your screen, and we will pass it over to you to begin your presentation. Thanks a lot for being here today.
Oh, okay. I need to escape and, right, 1 second. Share the, okay.
I'll just give a quick shout out again to those of you who are new to our scientific community for the first time. Visit us at gfi.org/community to stay up to date on seminars like these, networking sessions, etc., so that you can work in collaboration with scientists from all over the world to accelerate the pace of.
Sorry, can you see my slide?
Yes, okay. I can see your slide. I see the breeding project team slide.
Okay, so I need to go one more up. Come on. Oh, previous, and previous. Okay, awesome. I'm ready now. Sorry for the technical. So I'm still learning all this webinar thing.
As are we. Yeah. Awesome. Well, you're welcome to take it away. And thanks again for being with us today.
Thank you so much, Amy. And good morning, good afternoon, good night. I know because people are from UK and all over the world, and it is so much great pleasure to be here. And first, I'd like to thank the Good Food Institute, especially Research Director, Dr. Erin Clayton. Because of her, I got into the Good Food Institute research, and thank you, Erin, for inviting me to do this presentation. So today, what I'm going to talk about, how plant breeding or like the plant biofortification programs can influence the food processing, how we can link from food processing and the plant from the ingredients, basically farm to table. And by training, I'm a plant scientist. I also have a soil science background and also the agriculture background. And I worked at the Canadian Biofortification Program. And with my PhD, I start learning and working on the pulses. So in these pictures, you see our breeding plots of field pea. And as I said before, this is a talk about farm to table. So, and then we extract this organic protein. I have a team of people helping in the lab. And then how would that affect on the developing new food products? This project, not only the Good Food Institute funded project, this breeding project is a big one, which is funded by the NIFA USDA ORAI program. The breeding program, all done by Organic System. And I lead this project with the co-project director, Dr. Rick Boyle, because we are breeding the crops for the crop rotation. So Dr. Boyles, he's assistant professor at our department, Plant and Environment Sciences at the Clemson University, and he's the sorghum breeder. So with him, my program does the peas and his program does the sorghum. So it's nicely complement for the organic system. So because my crop provide the nitrogen and then it benefit the cereal crop as well. always I say that one person can do everything. So this project is done by many people behind the screen. I'm just in the front, but there are many people supported this program. So NIFA funded breeding program, we have the senior advisory team, Dr. Steve Krasovich, he's our APT Institute director and well-known genetist, bioinformatics and the genomics expert. So he helped on genomic side, and my two in-house breeders that I have, Dr. Shaip and then Dr. Kumar, he's the Icarda lentil breeder. And also we are very thankful to Dr. Maggie. She shared a lot of her breeding material with us to test in the organic program. And we are still testing her material, and she's a big advisor for this project. And then quite a bit of graduate students, my graduate students, the PhD students, and also field team. Without them, we won't be able to do anything. And then my phenomic lab, led by Adam Kay and Tristan Lawrence, manage my field. And also we have an experimental station personnel that helping with publishing and everything, and also the advisory team. I'm really grateful. With all these people, we have a Clemson Research Foundation, because I said this is farm to table. And then especially my husband, Dr. Pushparaj Thavarajah, he helped us with the food science part of the work, and together we submitted pattern. I'm going to talk about that a little bit. So Clemson University Research Foundation take care of all our IP for the breeding program, as well as the food product development. So let's talk about what is the critical need of organic agriculture. According to USDA 1995 definition, it is an ecological production management system that promotes or enhances the biodiversity, biological cycle, and the soil biological activity. So because it is a sustainable system and it is organic, we are not adding any pesticides or only chemicals or anything we can do, which is the USDA certified. So when you think about the broader definition of the organic agriculture, pulse crops are quite a nice fit into this system, because pulse crops fix their own nitrogen out from the air and provide phosphorus benefit. I have a PhD student who is graduating in 2 months and already found a genetic makeup for the phosphorus uptake and some of the special SNPs we identified for pulses. So these pulses are really nicely fitting into the organic system because of the biological benefit. And then if you're not from US, but I am from South Carolina state, which is quite a home for the 25,000 farms, totaling 5 million acres. And our farmers are not like Midwest. Our farmers are quite smallholder farmers. They're specialty crop farmers, and organic agriculture is quite fit into our cropping system. But if you look at this South Carolina commodity map, you can see there's a lot of corn, peanut, and other specialty crops, but that is not the pulses in it. So pulses are winter crops for here because South Carolina is very hot. And cool season food legumes like field pea, lentils, and the traditional sorghum like cereals like sorghum nicely fit into the this cropping system. So that is about South Carolina. Let's think about what is the global supply of pulse crops. I wanted to talk about 1961 to 2009. This is one of the data that we collected. This is because I want to talk about the pre-COVID. And you can see how much fat supply and sugar supply compared to the lentil supply. And there's a huge gap in between that. But with that, this publication came out in 2005 from Dr. Welch and Dr. Graham when they find the concepts like for the Harvest Plus. global micronutrient malnutrition. One of the hypotheses they set after the Green Revolution, it increased the cereal crop production to reduce the hunger, which is very good. But with that, it reduced the traditional indigenous pulse crops, which provide the protein and other micronutrients, which cause the micronutrient malnutrition or obesity today because of the lack of some traditional crops. So you can see here how much difference between the increase of production from the cereals and the pulses and the population. So you can see a different, there is a gap. So this may be the reason for the poor malnutrition, like the protein or micronutrient malnutrition. So what is the global protein need? Today, pre-COVID, even before COVID or after COVID, still we have billions of people affected by the malnutrition. Malnutrition can be micronutrient either or the protein malnutrition. And with that, also obesity and overweight are increasing. 1 in 3 is overweight or obese. So what do we need? Our food systems are failing, right? And because of the COVID, the transportation system damaged. But even before the COVID, our food systems are failing. That's why most of these unhealthy habits or less exercise, unhealthy food, that create this kind of problems. So therefore, we need to supply protein-rich whole food to the community. So our goal is to look at the food system approach. So when you look at the plant protein, there are challenges. One challenge is the plant proteins are lack of one or more essential amino acid. Proteins may not fully digestible. It's not like the animal protein. Plant protein has anti-nutrients, so it could be difficult to digest. Stabilizing the native protein and also structure. I am a great plant lover, so I have my smoothies and then I add protein. And when I add the protein, I do not like my smoothie separated. So it had to be structure, stability, and the functional properties. Also, organic is getting more excited about, people are talking more about organic proteins and organic food because of the chemical toxicity and the pesticide. And also the consumer preference is more important, color, texture, and flavor. So these are the more of the global needs when you think about the plant-based protein industry. So these are challenges for the plant breeders. Also, these are challenges for the food processing. So how do we narrow it down, these problems, to make a better protein? Now I like to introduce you about the pulse crops. So in the left-hand side of my screen, you can see all sort of beautiful food that made from our lab. Pulsars, also known as poor man's meat, because it provides cheap protein. Examples are field pea, lentils, and chickpea. And this is lentils, lentil curry, or like you could have chickpea salad. This is a soup that me and my husband love this soup for our lunch. And then we make this hummus. And if you wanted to add more organic protein into that, you could do that. Or rice and lentils, which is one of the leading food in the Asian countries. Especially I grew up in Sri Lanka, we eat 3 meals of lentils with rice. And also it can be used in the bakery products. So many foods like dal, soup, and many more. Pulse crops originated from Near East, and Canada is the largest producer right now for lentils. And the North American production actually begins in 1980s. But when compared to the organic production, it is really low in US. So let's look at pulses as an ingredient. In the left-hand corner, you can see I got the lentil as an example. And 60% of lentil seed made of carbohydrate, and it has 25% of protein, and the rest is the rest of the nutrients that you can see, fat, ash, and the water. So Why it is so important, even the carbohydrate, 40% is starch, 20% is prebiotic carbohydrate. From our lab, we proved that having a lentil-based diet in the rat, we had a rat study, we see that having lentil-based diet for 8 weeks even increased the bifidobacteria that reduced the molecular markers for obesity reduction. And then it has a 25 to 30% protein. So when you compare lentil, peas, and chickpeas, pulses with rice, you can see clear difference of protein, 20 to 30% protein in pulse crops and 3% of protein in rice. So this is why most of the poor countries, developing countries, we combine rice and pulses to get the balanced amino acid profile. It's not only protein, look at the other nutrient profile like iron, zinc, selenium, and at least 10% of iron is bioavailable. And not only that, beta carotene, folate, and lots of prebiotic carbohydrate, especially resistant starch, and there's a food processing methods also help. So what my point here is, I'm telling plant pulse crops are not only protein rich, consider it as a whole food. It can provide many other things. So with that, what is a quality diet? This is something we do as a professor and as in my teaching classes, like for the students, this picture that how we introduce the lentils and they enjoy it because some of the parts of the South here in US, lentil is not very popular. Pulse crops are not very popular in the state, but you have to introduce that. So when the WHO definition, what is a healthy diet? Healthy diet highlights the importance of starting healthy eating habits. So if you don't have pulses in your diet, you might have to include pulses into the diet in the early life. Then you get used to the taste, and also it limit intake of sugars and free salts. So it's low in salt and low in free sugar. So in this table, in my right-hand corner, you can see the essential amino acids you required in your quality diet should have all these 50 plus nutrients, including air, water, energy to all the vitamins, trace element, micronutrients, lipids, and protein. So that is the balanced diet. So pulses can provide many of these, including the proteins. So that's why the special about pulses, and that's why I love about pulses. So what is our sustainable solution? If you go into the Good Food Institute database, you can get all the new information about the market information on the plant-based protein industry. So I'm not going to put any of that information because it is available in the GFI website. You can see, but it's 9.5 billion by 2025, which is very impressive. So it is time for most of the plant-based protein industry to come in, and pulse crops are very important. North America has about 38% of the global protein market. So our goal is if we can reduce a little bit of soy protein market into the pulse protein. So that is one of the targets that we could look at. And we could go for lentils or peas or chickpea protein. And as I said before, pulses are high in low digestible carbohydrate, protein, and micronutrient. So it is a whole food. So that's a nutrition type. And what is the agronomical benefit? It provides nitrogen and phosphorus to the grower, so no added nitrogen fertilizer to the subsequent crop or to grow in pulses. Some soils you might have to add phosphorus. It's based on whether you add rhizobia into the soil or not. So pulses are very famous for most of the bread, snacks, and much of the food applications. So our approach is to have a nutritional breeding approach to increase the nutritional content, at the same time looking at the agronomic adaptability. It's not easy because most of these nutritional traits we're looking at not controlled by one gene or single gene. It could be more quantitative genes. So it is kind of difficult. So our approach, we call this as nutritional breeding approach. in early 60s, I'm sure you all know about Dr. Norman Borlaug, who is the Nobel Prize winner for the Green Revolution. He has done a tremendous job on breeding cereal crops and reducing the hunger, eliminate the hunger. But with the increase in cereal crop production, which is really good, and the nitrogen fertilizer application, which is good, increase the crop yield, we have more food for the hunger. But with the Green Revolution, it reduces the impact for the indigenous crops. So we are looking at a little bit more than Green Revolution. I always call it as a greener revolution that will provide not just food, we want more nutritious food. So our breeding goals to make sure the human health is the clear goal, and then improve the yield with nutrition quality, improve the access, utilization, and also the nutrient bioavailability. So in the right-hand corner of this food system approach that you can see in this diagram designed by Dr. Jerry Combs, one of the leading nutritionists from USDA, who happened to be my mentor. And then we have been talking about what is our clear goal before designing a nutritional breeding program. It had to be human health. Our outcome should be healthy humans. So the agriculture is one of the things within there. But if you see, it is combined with many other factors: environment, policies, economy, resources, agriculture practices, everything in that system. So we are building the food system. Improved health can be addressed by changing the paradigm for agriculture. So this is the bigger picture, how we approach to bring the human health into the breeding programs. This is like a nutshell of the bigger picture that I put into a figure. So we have three sectors going through. So the first is the plant breeding. So we select the germplasm, or we look for the germplasm and the varieties or anything based on the newly developed phenomic and genomic tools. And in this case, we are targeting the precision protein quality traits, but it's not we have to have appealing to the growers. So we have to make sure agronomic adaptability, disease resistant yield with the traits like precision protein quality. And in the precision protein quality, you can see this is our germplasm in the second picture. So we have a wider range of germplasm we are testing in South Carolina for the organic system to pick on these nutritional traits. So under precision traits, we are looking the germplasm for protein stability, native protein quality, isolation, and also texture, color, flavor, and aroma. There are many phenomic traits that we are looking at, and then we have to develop the phenome tools to make sure to select these things, because measuring only total protein might not be the best way to increase the protein quality. And at the end, if you develop the varieties or if you develop the ingredients, we have to test them for the end quality, especially amino acid balance. Is the sulfur-containing amino acid, is there enough? Lysine, cysteine, methionine, all of these are balanced. What is the digestibility? What is the digestibility test? So there's a lot of things that happen in our lab looking at the native protein structure and the nutritional value. So this is how we approach our system. And the bigger project goal is the long-term goal is to develop the biofortified organic FLP crops for organic cropping system. And the first thing we need to look at the adaptability to South Carolina cropping system, organic system, grain yield and agronomic adaptability. And then we look for nutritional traits like protein quality, carbohydrate quality, and the other micronutrients. And then I have a team I collaborate with to develop the tools for genomics and the bioinformatics. And then we do on-farm education and we do education for the consumers. And you can see in the right-hand corner, these students are preparing food, because that's the way we should start. It's not only growing crops, we need to teach them how to eat, how to use them. So it is important, the education. And our objectives here, these are the pictures from our breeding program. This is Dr. Shaib, my in-house pulse breeder. And we evaluate first the germplasm and the wide range of genotypes in the field. And then we pick these parents, and then we make crossers here in the greenhouse, and then we increase them, and then we put them back to the field, and we test them in South Carolina on farm locations. Also, we developed the inbred lines for genomic work, as well as most of the germplasm we will develop for the different nutritional traits. And then we conduct workshops, and also we teach courses. And at the end of this talk, I will share some information on if you wanted to learn more, and you can join our workshops. So let's talk about Pisum sativum, which is pea, also known as field pea or dry peas. And peas are grown and consumed as fresh or dry peas. We are working on the dry peas mostly for the human food or pet industry, mostly for the protein and the prebiotic carbohydrate enrichment. And these crops are old as 7,000 to 6,000 BC. It has 7 chromosomes and 2 colored flowers and white flowers. And we are mostly working on the white flower one. And the 2 cultivars that we work in garden peas and field peas. So our program looking at the mostly Pisum sativum dry peas. And the 2 different growth habits, and also they are annual crops. And in South Carolina, we use this as a winter pulse crop. And I'm going to share some data from 2019 and 2020, our field work we done in the organic on-farm locations. We have 2 locations in Clemson and Pellion for 2 years, and we tested 25 cultivars and 19 breeding lines from Dr. Rebecca Maggie's program in Washington State, and she's the conventional pea breeder. So we tested her lines into the our program. So we had the 1 acre of commercial pea lines. Because we grow that, then Dr. Boyles can go and put his sorghum advanced lines and see whether how much nitrogen benefit coming from. And then we rotated like in the fall and winter our pea lines in these farms. So the growing conditions, all peas were grown at the USDA organic certified land, fields were conventionally tilled, and we keep eye on the soils because of we want to make sure there's enough nitrogen. Basically, organic soils have less nitrogen compared to conventional soils, so we keep track on the nitrogen. And we did not provide irrigation because we wanted to have this low input crop. And the seeding rate and everything is in there. You guys can go on to our website. All the details are there, how we grow the crop. And we put Hampton, one of the Dr. Maggie's line, as the reference crop to test the adaptability. So you can see here the temperatures. The crops were planted in January, which is the winter, 3rd week of January, and we harvested in May. So right now we are getting ready to harvest our current nurseries. And what we did a lot of agronomic traits because this is a new crop for South Carolina, and we did a lot of agronomic testing, and we measure from germination to vigor to flowering dates and the maturity and all the agronomic traits that we could do. This is Dr. Maggie that checking on the lines, and this is Dr. Shaib and checking data. And we also use, we collect all the data using phenotyping apps. It's called PhenoApps, developed by Dr. Poland and Jesse Poland and Mike Go's lab. And it is very easy. We are not using any more pens and pencils and paper. So everything taken up by the Fieldbook app, and it create and we can customize however we want to collect those agronomic data, and then we export it into the Excel spreadsheet, which is very easy and less time consuming. And this is how it's looked like in the field. In the left-hand upper corner that you can see, that's the germplasm harvested. We have harvested by hand because it's a very small amount of seeds because of the genetic variation. Rest of the plots we harvested by using the small plot combine. And this is Tristan, my field manager. And then once we harvested, we dry it, it come to my lab, which is the phenotyping lab. We use quite a bit of different analysis method. So this is part of the project funded by the USAID Feed the Future Innovation Lab. So in this case, we take the nutrient flow as the protein quality. So we are testing our lines for methionine, cysteine, tryptophan, and lysine. So these are our four major amino acids that we wanted to measure. So we take a one seed, crush it, and then we don't do any chemical treatment. And we have this small gadget that FTIR, Fourier transform infrared spectroscopy. That's what we use to measure our protein quality and the total protein. And then we validate this data using model build using HPLC and the HPLC and LCMS that help us to build the validation models. And this is Ahmad, one of our PhD students working on the seed quality trait. So now we are in another step that we have a handheld unit that we could actually go out to the field, and with the model building, we could take reading before harvested in the dry seed. So we don't have to bring it to the lab. So this project was funded by the USAID because these tools will help for the breeders to screen their germplasm very rapidly. And that's the same tool we use in my breeding program. So let's look at about the, oh, I want to go back. So this is how the data look like. You can see the grain yield. PLP is a new crop, but yes, we can get 45 bushels per acre, just like the conventional. So the yield range is quite different. And if you see the protein is the same, we got the range from 15 to 30. And there are some lines are very good. These are lines that bred for the conventional system. And then we put them in the organic system and see how they perform. I think some of the lines perform really well, and we see the genotype difference, and then we see the environment difference. So this is the grain yield differences based on Clemson and Pelion locations. You can see this is the location different. And then what is the relationship between the grain yield and protein, which is everybody knows, it is a negative correlation, but we can further improve the total protein if we need. So you can see here is the yield range from 500 to 3,500 kilograms per hectare. And then here the protein, we range from 15 to 35. So it's a nice combination, the varieties available right now. So we have a hope that we could increase the protein. So are we going to increase the protein content, or are we more interested on protein quality and the digestibility? So these are the things, that the objectives that you have to put in your breeding program to improve these varieties. And let's say the other nutrients, like I said, the protein range from 12 to 34, which is exactly same as the conventional range, little bit low. I would expect little low nitrogen content in the organic soil and low protein in the organic system, but our farms were managed really, really well. These are farmers' fields. They were managed very well. And then that's probably why we see quite a nice range. And then we are not only protein, we are interested on prebiotic carbohydrate like sugar, alcohol, simple sugars, RF4, raffinose oligosaccharide, starch polysaccharides, because those help to improve the gut health. And then if you're a breeder, you will ask like, oh, all these nutritional traits that you're measuring, are those heritable? Some are heritable, some are not heritable. So we calculate the heritability estimate protein for these subpopulation of these cultivars that bred for the conventional system has low heritability estimate for protein. So we have a lot of work to do, but the sugar alcohols and the other sugars, we see range of heritability for these traits, which is a good sign. So we can incorporate this information into the breeding program. And the same thing with the other nutrients, like we looking at the yield and how it affect on the minerals and the phytic acid. Those yellow highlighted ones are the best ones that you can see how they perform very well and very good, suitable for the incorporate that growing piece. And these are from the results now in the South Carolina. Growers has a choice that they could go pick any of these lines, promising lines that they can grow right now. So these information is available in the website, which is with the high protein and high yield. These are the good varieties that they can grow in the organic system for the moment. And so that's the breeding side. So now I wanted to move the switch gears and come into the food processing side. So how do we link this to? This is complicated. So first, we need to do germplasm selections for adaptability for South Carolina with the yield and other disease resistance and things like that. And then what kind of protein quality traits that you want to incorporate. So in my case, I like sulfur-containing amino acids and lysine and tryptophan. And if I can measure protein digestibility, color, texture, aroma, and flavor. So once we know these things in the germplasm, we can actually develop the genomic resources with our genome team. And these resources will be available for the breeders to select using marker-assisted breeding. And then that ingredient that we start. So also we can use these ingredients or the varieties, how the protein isolation and blending happen. Is it increase the native plant protein structure? Can we keep it? What is the functionality when you blend it? How it's that effect? So also nutritional traits. And what ultimate goal is, we wanted to make a better human health, better protein for better nutrition quality, better human health. So that's our target. So with that, let's see what are the food industry challenges. So when I talk to the food industry people, their challenges mostly food processing and nutritional challenges. Sulfur-containing amino acids are basically low in pulses, so that also challenging for their blending and other processes. And not fully digestible, and the anti-nutritional components, and also having thermal degradation and also chemical toxins. So growers, organic growers has a problems because they don't have cultivars that only bred for the organic system. Right now they're using cultivars that is bred for the conventional system. So in the organic industry that we have problems with the developing, or we need to develop cultivars that's suitable for system, and then also food processing industry. So I put it as a line figure. Right now in the commercial protein, some protein has very high amount of sodium, and which is not good for hypertension patients. So, or it can create some health issues. And then other issues are low yield, 50 to 60% when compared to the organic protein, some of the protein extraction issues. So there's a, regardless of how you extract the protein or how you increase the yield, you will concentrate the chemicals and the other pesticides. So that also another nice thing about having organic protein, that we have less chemicals and less pesticides. So with that, my team, we submitted a provisional patent. This is fully funded by the NIFA and the Good Food Institute. And we submitted the invention, organic plant protein, and the patent number is there. It's a utility patent about how organic protein extraction. So we did this for organically grown pea and sorghum both, and no chemical residues. And it has the balanced protein by blending them. and then it is highly digestible, enriched with other micronutrient and also prebiotic carbohydrate. And we have 100% USDA certified organic protein, gluten-free, and it is non-allergenic. A little bit more about the profile of this protein. This protein was extracted from our key variety that we have. It's from a bulk. It's not from a one variety. So it's from a bulk organic ingredient that we tested in our field. That you can see that we had organic pea, organic sorghum, and then we have a control, which is the store-bought protein. And you can see the protein percentage in these extracted protein peas were higher, 74%, and sorghum has 65. And then we found quite a bit of other prebiotic carbohydrate, and minerals wise, our ones has no sodium, and we have high in other minerals compared to the control. So it is 99% organic protein, and we are very excited about it. We filed the patent, me and my husband, and then if you're interested, you can talk to Clemson University Research Foundation to look for more details. And then how the cultivar versus our extraction procedure of the organic protein isolation different. This is why I'm saying the ingredient matters. So when we look at the different cultivars, we had two processing methods. We call one as the mild and the other one as the harsh. You can see about 80 to 85% protein in the extracted protein. And from the cultivars, it's quite different. And so it means that even for the isolation process, genetic matters and the environment matters as well. We haven't done more on the environment analysis yet, but we are hoping to do that within the next couple of months. So this figure shows most of the common cultivars, they have different response to our isolation process. So why organic protein? Now you can see this is the smoothie that we want to make without having no separation, and we want to keep the structure. And this is our extracted protein in the left hand corner. So 100% organic protein, and we discovered two processors, and they both works well, and no sodium, and also especially no chloride in the isolation process, no chemicals, no pesticide, and flavor. We're still working on the flavor, and we don't know exact target compounds yet, but very low beanie flavor compared to the store-bought one. And then it has low levels of phospholipids, and we assume there's no pork-like meaty flavor yet, but we are still working on this. We will have consumer panels, and this is in the middle of the process, but I wanted to show you how the breeding and food processing linking, and this is the provisional patent that we submitted. And what are the food applications that we are thinking of? We are thinking of like the Impossible Burger or Beyond Burger. Maybe we could use this organic protein as a substitute to the soy protein, and maybe nutritional supplement and human food, animal feed, and it is pure organic. So there is much more work to be done still, but it is promising. And going forward, I'm very pleased with the NIFA, USDA NIFA that funded grant, that now we can say South Carolina can officially grow organic peas without much problem, but we have a lot to do for the diseases and things like that. That's why the breeding is important. We have to continuously monitoring. Eventually this crop going to be a better crop, but we need to do other things like disease control and things like that. And also we are testing a lot of genetic material for the adaptability as well as the protein quality. And also we need to test the planting days. This is only 2 years this breeding started, so there's a lot to do, and we planted nurseries and things which is going forward right now. this is the one I wanted to tell you. So if you are interested on learning more about organic agriculture, breeding biofortified pulses and grain, you can join our free webinar or the free organic plant breeding institute. This happened pre-COVID. We decided this on campus, but now because of the COVID, we are doing online. It's the 5-day sessions that we will have hands-on work. And I encourage you to look for it and register. It's free. And if you need more updates on our work or our tools, this is the website that you have to go check on. And finally, I like to say that at Clemson, we deliver skills and insight that will contribute to the global food and nutritional security. We are not only playing football, we are famous for the football, but we are also really good at what we do for the global food and nutritional security. It's a beautiful campus in a very sunny day, and it's hot. Very rarely we get snow, so that's why pulses grow well in here. So I won't be able to do anything without the funding support. So my lab is funded by USAID, ICARDA, Clemson University, USDA, NIFA. We are very fortunate for the OREI program that supported the breeding program and the Good Food Institute. Nothing possible without the state government, South Carolina Department of Agriculture. All this breeding started from a very small grant that they give to test these lines. I've been in Clemson for 7 years, so this is what, and then all these people, especially the breeders from Canada, Dr. Walkington, and Dr. Bing, and Dr. Maggie, they were so helpful. And our pulse seed companies, Meridian Seed, USA Pulse, all of these people helped to get this program built and do a good job. So that's all I have, and thank you so much.
Thank you so much, Dr. Dil Thavarajah. That was such a fantastic seminar. I learned a ton and really excited to delve into some of these questions. So it looks like we have 11 or 12 questions here in the Q&A section already. And I can go ahead and read some of them off. We'll spend approximately the remaining 40 minutes or so on Q&A. For those of you who have not yet dropped questions into the Q&A section, please feel free to do so, and we'll get to them if we have enough time. All right, so one of the biggest questions I see kind of raised here in the chat by several individuals is this concern around the growing global population, and with it, the skyrocketing rate of protein demand, which you touched on towards the beginning of your presentation. So since we know, the population will increase and the market for plant-based protein will also increase with it, what do you think are some of the best strategies to keep up with plant-based protein production? How do you balance the relative advantages and disadvantages of organic farming versus genetic modification, for example?
This is like a question that I get all the time about my simple answer, we need to have a balanced approach. We can say this is better than this is better than that. No, let's go with the balanced approach. Organic is, yes, it's time consuming and you need to have a really good management system, and it's sometimes it can be expensive too. But it is for a niche as well, but it's help for the soil. So that's my thing for the organic, and it's really good that we use less pesticide. But we cannot practice organic agriculture all the time. So we need to have a conventional breeding as well. So what my thing is, I think we have to optimize everything we have. We need to get everything into the table because we have to feed the people. In 2050, this 10 billion people, we need to have approaches. I don't think by doing organic only we can do anything. We have to do everything. So let's think about the multi-purpose approach. So yes, it can be pulses. It could be any other sources of protein as well, because animal protein might not be able to feed for all the people increasing. So we need to have a sustainable approach. Pulse crops are really, really good. So I only talk about peas and lentils, but there are many other pulse crops that in India, if you go, there's a lot of pulse crop, pigeon pea, tur dal, many pulse crop, mung beans. So we need to invest on those indigenous crops for different countries that to improve it. And not only pulse crops, we have to have a protein, yes, pulse crops and many other, some cereals as well, like even sorghum protein is very good. So we have to invest on those indigenous crops as well. At the same time, we need to think about how do we bring the food, maybe invest on vegetable roots, all the cropping system to have a balanced diet, not only protein, because human can live only having protein. We need to have everything. That's what my take home message, that with that table, what I'm showing, you need 50 plus nutrients. So think about giving all the things as a whole food. That's how I approach genetic CRISPR, the genetic editing, GMOs. All these technology is important, and we can learn from that, and we can use those technologies sometimes to learn things in the organic agriculture as well. We don't have to use it, but we could use that technique to learn the function of a gene. That is so important to incorporate that, incorporate in that to understand how the genetic makeup works. That's how I take it. So I'm not taking one side. I think we should do all and work together to get this going. That's kind of broad answer.
Yeah, no, I appreciate it. And I do want to, signal boost a couple of the things you said there. I think you're absolutely right that we need to be diversifying our protein sources. And there are so many underutilized crops that we haven't looked at. And I also agree with what you were saying about the fact that sometimes the conversation around protein is really reductive, or the conversation around feeding the world is reductive because it isn't just about protein. And there are so many other nutrients that we need to be looking at as well. So thank you for your response there. I do have kind of a related follow-up question related to at least the nutrition of legumes and cereal pulses. So from Mary Lise, what about the anti-nutrients in legumes and cereals like phytates and bison? Do you see this as a problem in plant-based foods? And do you think that it's possible to mitigate or decrease the risk of anti-nutrients through breeding practices?
That also a very nice question, and I always get that question as well, because pulses, plant-based, and then anti-nutrient. This is something we always talk about it, like phytic acid, tannins, those can, especially phytic acid, that can bind with iron and zinc, and then it excreted out from the body, right? That's a problem. So that's why people concerned about anti-nutrients. So I would say this. So now I know for sure that the peas and lentils has low concentration of phytic acid. And also phytic acid has many different forms. The final form is IP6. In the plant, it starts from IP1, inositol phosphate 1, 2, it go from 3 to 4, 5, and 6, based on how much phosphorus group add into. So one of my PhD students just completing her PhD, and she's starting as a postdoctoral scientist in Missouri. Her project, she looked at the pea germplasm, and she looked at how much phytic acid in the germplasm. Actually, this is really low. I'm not very concerned to reduce it, because we wanted to have some level of phytic acid in the germplasm, because that's the phosphorus storage for the babies. Like if there is not enough phosphorus, we will not have healthy plant. So we need to have phosphorus in the plant, certain amount, to develop the plant system, just the plant. So by looking at the genetic diversity, now we understand the genetic makeup of phytic acid. We even know the significant genomic regions that responsible for this synthesis. So therefore, I am confident about the peas what the range we have is suitable. We don't have to reduce it. I want it to keep it the way it is. And the same thing with lentils. Lentils, I don't know much about genetic makeup yet, but we see in the germplasm from ICARDA, when we look at it, it's actually even lower than field peas. So I wouldn't go there and knock down the genes or do anything to reduce it. I want it to be as it is. And the other legumes, we have to look at it, and we wanted to know it's certain ranges, like we want like about 100 to 400 milligrams per 100 grams. So it's quite low. And I also know about mung beans. So I'm not that worried about phytic acid. Tannins, beans have a lot of tannins too, so we haven't worked much with the tannins, and that can be an anti-nutrient. For the moment, phytic acid is not that something you have to worry about. So that's a good thing. And also our isolated organic protein, if you remember that, we see it and it has some amount of phytic acid, and we are not worried about that. They are very low concentrations. So if it is very high, we are worried about. At the moment, I'm not very much worried about it.
Awesome. Thank you so much, Dil. Here's a question from Zach Weston. With a lot of different seed lines, breeding companies protect their IP with patents and by making it difficult or impossible for farmers to save seeds from year to year, which provides a really important financial incentive for seed breeders, but makes it a lot harder for farmers. So the question for you is whether you think pulse breeding advances can be protected via patents, and well, can pulse breeding advances be protected via patents and put barriers to seed saving in place?
Well, so our Clemson University, my program is a public breeding program. So we have a IP protection done by the Clemson University. We do the variety release for the public. So so it's depend on the individual who work in different institutes. So mine is very public, so I don't think that is going to be a problem. And all that, I totally agree with you and like that comment, because when we also exchange our germplasm, we will sign MTAs and all the IP agreements. That is happening right now. Even I work with the seed companies. I have to sign everything, and we do all legal process by the Clemson University. It can be a problem. So I don't know how we handle that. That is kind of beyond my comprehension. But one thing I know, my breeding program is public, that I wanted to make sure that varieties that we develop, that should be available to our growers. Our first priority is our seeds available to our growers. So that's the Clemson University. And if you need more information, you could approach CURF, Clemson University Research Foundation. They have all the procedures. So I totally understand. I totally agree with all this. And I go through that too. So that's effect on the farmers. So if we can reduce that barrier, I want to make sure my farmers get the seeds.
Awesome, thank you. Yeah, it seems like maybe this is an ecosystem level white space for people to think a little bit more about how can we facilitate the reduction of barriers to seed saving year over year across the ecosystem. So here's another question around improving the nutritional quality of pulse crops from Ite, who says, this kind of work is really important, and you've given a great seminar here. But how many years approximately does it take your breeding program to release a new nutritionally improved variety?
2024.
So does that, yeah, how many years of research?
So another 4 years, four and a half, 5 years, I think I will have a couple of germplasm ready to release. I wanted to test several years in the on-farm locations. So our target goal is to release the first one in 25. So it's like another 4 years. We do see the increase very rapid in in a year, like a speed breeding method. But I'm hopeful because my program is only 2 years old. So we are hopeful and we have plans, and so far we are on the time. And yeah, the first protein one, actually, we were thinking 24, 25.
Awesome. Super exciting.
Very exciting, yeah.
Yeah, so I have a quick question about whether you can, or there's a question here from an anonymous attendee about whether you can elaborate a little bit more on how you quantified prebiotic carbohydrates.
We used high performance anion exchange chromatography, and you can buy those system if you want from Dynex. So that's the company that made that. So we use whoever that can email me, I can send the procedure and the papers. So if you go on my website, you will see the all papers. We have the methodology. So it's like a extract the seed, we grind it, and we have extraction protocol, and then we use the high exchange anion exchange chromatography to measure it. Yeah. So it's like a routine procedure. We do it in our lab. And then that will HPLC we use only for the low digestible carbohydrate, and then you can measure starches and resistant starch, amylose, amylopectin using enzyme assay method. So you could buy the mega enzyme. We don't buy mega enzyme, but we develop our own assays. So those protocols are in our published methods. So if anybody interested, just shoot me an email. I can send you. So those things happen every day in the lab.
Awesome. Thank you so much, Dil. Here's a question from Sajid around protein isolation. So the isolation of proteins from different kinds of plant seeds is always so very challenging. Do you have any suggestions for isolating proteins in as pure a form as possible?
From what? From the seeds? I really don't have any good answer for that. So we struggled a lot. And then finally we came up with the method. That's the one we patented for the isolation procedure, because organic protein isolation is more difficult because we cannot actually use the heavy chemicals like the conventional use. So I really don't have much of the answer for that. But if you send me, probably I could talk to my food science team and get some idea, depend on the what crop you work, and maybe we will have a little bit of background idea. Yeah, we work very much on the piece for last 2 years to get optimum method, because I was the one of the first grantees from the Good Food Institute, but we did not actually finalize our method for 2 years, and we need at least another 2 more years to purify it, right? And then make it much better. So, but we have a really good method. We work very hard on that. So, but depend on the crop and thing, maybe we could revisit it. If you can email me, and maybe I'll think about it, and I talk to my husband as well.
Sweet. Sounds good. Sounds like there's room for additional discussion.
Right.
Here's a question from Sajid, who's hoping you can speak a little bit more about the relation of your research to plant-based meat analogs. So what of the plant protein sources that you've worked with, which sources have you found are the most preferable for formulating plant-based meat, and what strategies, if any, do you think can be used to enhance the functionality of plant proteins to make them better for plant-based meat?
So we haven't worked with the plant meat analogs. So we just extracted the protein just for the food industry, just for the like to bake a cake, or like mostly my target was for smoothie industry, because we love smoothies. Because this started because I cannot eat any animal protein products, so like, and milk products. So that's why I use plant-based protein for my smoothies. So we haven't done anything with the meat analogs, but I think pea protein, you could try it, because I have a feeling the way that the functional properties work, how when we isolate it, I think it could be a best for the meat, but I don't know. We haven't tried that. And peas and lentils in our procedure, both peas, lentil, and chickpea, all the pulses that worked really well for the food in like the food applications like baking, smoothies, yogurt, juices. So I don't have answer for that.
Thanks, Dil. Here's a question from Malika, and I'll admit that I'm not positive what the technological foundation is of this question, but have there been any efforts in first growing crops organically and then externally injecting or infusing enzymes that lead to a higher production of protein content?
Say it again. We are injecting enzymes to enhance the
Higher
production
production of
in
protein.
the organics.
Mm-hmm.
No, we can't. In the organic production is very controlled because every step we do is under USDA organic certification. So if you do anything other than what listed under the certification, our certificate will be gone. So we are no longer organic certified. So in my knowledge, for our experiments, we did not add anything. We are very careful. And even when we use our equipment, we make sure we clean twice, 3 times, because if it is used in the conventional field, even our plot combines and things like that. So we are very strict on the USDA certification. So in my knowledge, I don't know. We haven't added or spray or anything. It's all natural. But there are chemicals and that USDA certified. So maybe those enzymes that she's talking about, maybe USDA certified, because there are like chemicals to control bugs, and there are neem products for their insect control and the biological controls. But I haven't done anything. Our crop is very new and we are learning. We use the biological controls mostly. We take care of the bugs to take care of the crops, and also like diseases we are still learning. And we could spray some of the chemicals which is USDA certified, but I don't know anything about enzyme adding to enhance the growth, which I haven't done.
Awesome, thanks, Jill. Here's some questions around other kind of fast-paced plant breeding companies. I guess this is just an overarching pulse check from you on what you think about the companies that are touting, 4 to 6 years to develop new strains and plant variants, and claim to operate on a much shorter timeline.
Can you explain me
Yeah, it seems like they're just asking for your opinion on the very fast-paced plant breeding companies.
I like that. No, I like it. If we can make it faster, so even we do the speed breeding method to get a couple of generations in a year, using all the greenhouse technology we have. I'm all pro it. I like it, but yeah, I don't know how. It's all about funding, right? Money, and it's expensive. So convention, anything you can release, but I would test out in the farmer's field. That is more important for me because I want to make sure the lines that I release, it's going to be helpful to the farmers. So I would test. It can be released. It can build rapidly, but we need to make sure these are right lines for our farmers. That's how I think.
Awesome. Thank you so much. Well, we have 15 minutes or so left on the clock. Dil, there are a few questions here in the chat or in the Q&A section if you want to take a look through and see if any speak to you that we can get to. Those of you who haven't yet gotten the chance to pose your questions, please do feel free to drop in and add some of your questions in. But you've touched on several of these in different capacities. I'm not sure if you have more to speak on them or not. For example, there's a question here from Jiao. who would love for you to elaborate a little bit more on your use of bioproducts like symbiotic bacteria or other biological products to increase the yield or quality of a harvest.
So we use the biological products for the rhizobium as an inoculum when we planting, because we cannot do any seed treatments because it's the USDA certification, only chemicals that we can use. We haven't used any other biologicals yet, because we wanted to develop these crops as a low input crop, and then we want to know how these crops adapted into the natural organic system. So I was actually looking for more diseases and pests and things like that. Like, we had quite a bit of aphids at the beginning of the crops. So when we plant, these are winter time, so we don't have very many bugs, but these peas start flowering sometime in mid March. So that's the time we get some aphids and thrips. So it was a nice observation. So aphids actually go away after a couple of weeks when the ladybugs coming starts. So it's like what we were looking for, a natural system. So it's depend on, and then depend on what type of crop rotation they have. So first 2 years, by putting these things, we haven't used any biological control, any chemicals, nothing, because we wanted to study and we wanted to see whether it's going to be adapted to the organic agriculture. Because there are a lot of things, but I don't want it to pollute right away, putting everything, and then we don't know a lot of unknown variables. And first we need to study, this is a new crop, so we haven't used anything. But I see some of these automatic biological things happen, and depending on, like, I think we will have quite a bit of diseases coming with the wet weather and the humidity. After April, the temperatures are going higher, so that's why early May that we could be able to harvest it. And for the peas in the conventional system, desiccation has to be done. There are organic desiccants, but we haven't used it. With increasing temperatures, like if I have like 80 to 90 degrees for temperature that high, we go in south in sometime in the second week of May, 80 degrees, 75 to 80, that will burn out the crop. So, and then it's easy. So I don't have to use any biologicals to harvest it. So that's the protocol we use right now. But we are also learning because it's new. But with the time, we should be able to have more precise methods developed. And all of you know that organic is very complicated. From the land preparation to the harvest, weed control is really difficult. So if you're not top of everything, you're going to lose your crop. So that's why most on-farm locations are really good, because farmers look for the productivity. And these farms are small scale farmers, not like Midwest, very large farmers. So my on-farm locations were really nicely managed and clean, so we did not have much of the diseases and things like that. Crop rotation is important too. So I am trying to go away from chemicals and see how much we can go with the natural ecosystem. I'm kind of optimistic person, so we'll see.
I share your optimism. There's one last question here from an anonymous attendee. Of course, a lot of folks in our scientific community, because we're focused on facilitating the shift away from industrialized animal agriculture, really value sustainability. And a component of a solution in service of a more sustainable world from an agricultural perspective is this idea of like a circular waste economy. This idea that we might be a little bit more resourceful in utilizing agricultural waste streams to feed the world or do other stuff with. So the question from this attendee is whether you have some thoughts on what kinds of byproducts can be used from peas and the pulses that you've been working with, similar to the ways that we use like corn and soy, but maybe beyond the standard, animal feed type of applications.
Oh, so it's like biofuel kind of area. That's what So the most of these pulse crops are for human food, like because it's a protein and all the micronutrients. So when you get the pulse seed, some, like for example lentils or peas, you could dehull it. You could remove the seed coat. Actually, when you remove the seed coat, some of the anti-nutrients, like you were asking, like some of the polyphenols, tannins will be removed, because seed coat has those. Plus, seed coat has really good stuff like cellulose and hemicellulose, dietary fibers. So we remove those. So most of that goes to the animal product, but and then the rest goes to the human. And I think if we used our crop for biofuel or anything, like, I feel like it's kind of waste, because this is like a really small biomass produced crop. Um, it's more designed for food or feed, the waste. And then you don't need to do very many techniques. You could like get the pea seed and throw it into a water pot and boil it, and you eat it. You get the whole goodness, like you whole enrichment of the nutrients. You don't need to break it into the ingredients. So like I develop for high protein work, but you really don't need to break and get the protein even out. But yeah, food application, we need to get the protein out. That also, like, we are breaking the seeds and take ingredients apart. That's true, but that's good for the different industry. But I don't really see that pulse crops go for the bioenergy type, but cover cropping is one of the other things that we could do in our germplasm, that there's a quite a bit of range of diversity we see for the cover crops. So cover crop is good for the organic production or reduce the nitrogen fertilizer application, because the biomass has quite a bit of nitrogen. So I would argue that instead of we make bioenergy or something, we could use this as a cover crop, and put back to the soil and enrich the soil, organic manure and things like that in that route, build the soil health. I'm thinking on the organic ground. I don't know, because I too much focus on organic system. But the same thing with the conventional system too. Cover cropping, peas are used as quite a bit for the cover cropping, faba bean, and in our organic farm we use faba bean as the winter cover crop. Um, and then the summer we grow peas, like the cowpeas and things like that. I don't work on those, but we can use those as the summer pulse crops.
Great, thank you so much. For the person who asked the question, I do think you could be curious in checking out Dr. William Chen's work at Nanyang Technology University in Singapore. His
Okay.
work is focused on utilizing spent grains to actually replace yeast culture medium. So as a enabling technology for fermentation that can then be applied to alternative proteins. So we do
Oh,
see this expansion of kind of creative applications for spent grains or pulses or agricultural byproducts.
sounds good.
Yeah, well, thank you so much for joining us today, everyone in this community, and also Dr. Thavarajah for imparting her expertise and wisdom. Before we wrap up today, Dil, was there anything that you wanted to say to this community of alternative protein scientists?
Um,
On the spot.
on the spot. I think this is an exciting time, because of the COVID, we need quite a bit of different food, especially meat industry has infrastructure and transportation issues. Luckily, we have vaccine, but there's a lot of people in Asia, like we know that India, like my friends are suffering, and like so pulses and other indigenous crops. This is our future, and this is our time to grow more and think about sustainability on the planet. So this is a good time to have more plant-based protein. And also we want to reduce the diseases and things like that, and having less chemicals also. So that's why I love about organic, but I think we need to build more food conventionally, organically also. And the plant-based work is fantastic.
Awesome. Thank you so much, Dr. Dil Thavarajah. Yes, there's definitely this opportunity to kind of bolster the resilience of our food system through exploring plant-based proteins. I really appreciated having you here with us today. Thank you again for your time. To those of you there in the audience, be sure to tune in next month. We'll be joined by Dr. Glenn Gaudet of Boston College and diving into some of his research on using decellularized plant materials for scaffolding for cultivated meat applications. So again, really fantastic to have you here, Dil. I hope everyone has a fantastic rest of their week, and we will see you again soon. Thank you.
Bye. Thank
Bye.
you.